Our protocol standardizes methodologies for sampling the gills of gravid freshwater mussels and characterizing the different stages of larval development, while also providing an online database for data submission and exploration of the Gravidity Almanac. The technique is non-lethal and does not substantially affect a female mussel's ability to remain gravid after sampling the gills. Therefore, the method can be applied to both common and protected species.
This methodology can be applied to organisms characterized in the Bivalvia families of Hyreidae, Margatiferidae, and Unionidae, which are distributed in freshwater ecosystems on every continent except the Antarctica.
It is important to open mussels with care, collect location information, and make sure data are collected properly. If identifications are questionable, one should take pictures and contact the appropriate experts.
Start by collecting live mussels from the field using tactile visual methods. Evaluate female gravidity by visual inspection either during or after collection by gently prying open the valves just enough to look inside and see whether the gills are inflated. If using a preserved specimen from a museum, perform visual inspection by opening the valves and inspecting the gills. To sample the gill content, prepare a 1.5 milliliter plastic microcentrifuge collection tube with approximately 1 milliliter of either water or ethanol, depending on how soon evaluation will take place. Unwrap a sterile 30-gauge bevel-tip needle on a 10 milliliter syringe, unscrew the cap to expose the needle, and push the handle of the syringe down so the stopper is at the 0 milliliter line. Pick up the gravid female and gently pry open the two valves using the